Hello everybody. Today I will be going over different iron uptake mechanisms found in bacteria. I will be covering both gram-negative and gram-positive bacteria and the different mechanisms these use. There will be quite a bit of crossover between the mechanisms used, or some differences being the methods of getting iron across an outer membrane and an inner membrane for gram-negative will be slightly different than the mechanisms used to get iron across a cell wall in gram-positive bacteria, but we will cover that when we get to it. So, starting off, we will be looking at gram-negative bacteria like Pseudomonas aeruginosa. As you can see in this figure right here, it gives a general overview of some of the methods that we're going to cover. Of course, you have things like siderophores, molecules that are secreted by bacteria to bind ferric iron in its 3 plus form. You have methods of transferring hemoproteins using hemophores, and you have the Pheo system, which is a specialized system that is used in anaerobic environments where ferrous iron is present in its 2 plus form as opposed to its 3 plus form. So as you can see in this figure again, the methods are pretty similar across themselves. The iron is taken from the environment through a receptor, through channels in the outer membrane into the periplasm, where it gets pretty standard across the board. The iron is bound to a chaperone protein of some sort, transferred towards the inner membrane, where it is passed through these permease proteins and this ATP binding cassette protein, where ATP is required to move this iron into the cytoplasm for the bacteria to utilize. Moving on to siderophores themselves, these are molecules consisting of R groups that give specific siderophores their characteristics, nitrogen and oxygen, which is responsible for binding to the iron itself. These siderophores are produced within the bacteria and secreted out into the environment, where they find ferrous iron in its 3 plus form bound to oxygen within the environment itself. The oxygen present on the siderophore will break those bonds between the iron and the oxygen and bind to the iron in its place. This will form a chelate. This chelate, this siderophore iron complex, will then move to the bacteria, or it will be picked up by the bacteria, where it will bind on a surface protein called the TUN-B receptor and be transferred into the periplasmic space through a protein channel. Once the siderophore is within the periplasmic space, it will be bound to a chaperone protein, like we said earlier, and then brought to that permease channel to pass through the ABC channel into the cytosol. Once the complex is in the cytosol, the iron will be released from the siderophore to be utilized, and the siderophore has the opportunity to go bind to more iron and keep the process going. Heme intake for gram-negative bacteria is a little interesting. Hemoproteins within the environment, proteins that contain heme, can either bind directly to the PHUR receptor on the outer membrane, such as hemoglobin as its entirety binding to this receptor, or the bacteria can release an HASA protein into the environment, which is a much smaller protein that will find a hemoprotein, bind to one heme molecule within that protein, and break it off from the whole. And once that HASA protein and the heme that it is complexed with is on its own, it can bind to a separate HAS surface protein and bring just that one heme molecule into the periplasmic space. Whether it is the entirety of a hemoprotein or just the one heme molecule in the periplasmic space, it is it will only be the heme that moves on into the cytosol. So once the hemoprotein is in the periplasmic space, it is degraded into its heme constituents. Once the heme is within the periplasmic space, it of course binds to a carrier protein and is transported into the cytosol, once again through the ABC channel. Of course, the heme, once it is in the cytosol, is not just entirely made of iron, so it is degraded within the cytosol into its constituents biliverdin, carbon monoxide, and ferric iron to be utilized in whatever way the bacteria needs. Some of you may have worked in a microbiology lab where you had to culture bacteria on blood agar, which is what this figure down here is showing. This is sheep blood agar that has been cultured with Pseudomonas aeruginosa. This is just when the cultures first grew. Later on, such as in this picture, you will notice that the blood agar becomes yellow, transparent in some places, around the cultures. This is a process called hemolysis, and Pseudomonas aeruginosa is capable of producing these enzymes called hemolysins that they secrete into the environment. And these hemolysins are capable of binding to erythrocytes, or red blood cells, and breaking them down into hemoglobin. So this is just a way that the bacteria can take bigger things from the environment and break them down into smaller pieces that it can use to bring more iron into its system. Moving on to the phyosystem, which, as I said earlier, is that specialized system for anaerobic environments where there is more ferrous iron and not ferric iron. Since it is an anaerobic environment, there is not quite as much oxygen present for the iron to bond with and reach its 3+ form. So the 2+ form that is present in these environments is soluble in water, more so than its 3+ form. And given this solubility, it is actually able to pass through porins within the outer membrane through diffusion without any necessary proteins to bring it into the periplasmic space. Once that 2+ iron is within the periplasmic space, it isn't actually able to diffuse through the inner membrane into the cytosol. So there is a permease complex comprised of the phyO-B, A, and C proteins that can take that 2+ iron from the periplasmic space and bring it into the cytosol to be used. Figure here, these PCA and piocyanin molecules are actually molecules that are produced by the bacteria itself. These can be secreted in the case of an aerobic environment to bind to 3+ proteins and reduce it to its 2+ form. So the phyosystem is not used solely in anaerobic environments. It is used to bring 2+ iron into the cell when it is present. So if there is an overabundance of 3+ iron and it wants 2+ iron to use, it can release these molecules to reduce that iron and keep the cycle flowing. So those are some mechanisms that are common in gram-negative bacteria. Let's look at some that are used in gram-positive bacteria. As I said earlier, there are quite a few similarities between the mechanisms that are used. There is quite a bit of overlap that is used between these as well, but there are some differences, such as these membrane-anchored binding proteins present in gram-positive bacteria that bind to molecules carrying iron on the outside and help pass them through the ABC channel, similar to chaperone proteins within the periplasmic space, but they are just present directly on the surface of the cell wall. We will be covering the ISD, the iron regulated surface determinant system present in these bacteria, which is a transport system used to move heme that is specialized to gram-positive. And we'll also look at transferrin and lactoferrin interactions, which are non-heme but still iron-containing proteins. So they're not typically part of a larger constituent. They're proteins present on their own that bind to iron. We are going to look at the ISD system first. Within gram-positive bacteria, this is similar to the heme binding systems found in gram-negative bacteria. However, in gram-positive, in the ISD system, it takes the whole of the hemoglobin at once. It has no specialized proteins to break off a heme group. It takes all of it at once and binds it to the surface of the cell wall. Once there, it it breaks down into the heme constituents and passes them through the cell wall using chaperone proteins. Once the chaperone gets through the cell wall, it will pass the heme group through an ABC transporter into the cytosol, where it undergoes the same degradation into belaverdin, carbon monoxide, and ferrous iron to be used by the cell. Transferrin and lactoferrin are a bit interesting in their interactions with bacteria because transferrin and lactoferrin are iron carriers present in blood serum within the host. They aren't produced by the bacteria, they aren't brought in by the bacteria, they are present in the host before the bacteria gets there. And these carriers bind to iron to transport around the body wherever it needs to be, and lactoferrin specifically sequesters iron away when it is not needed. In times of stress for the body, catecholamine hormones such as epinephrine, norepinephrine, stress hormones, can bind to these transporters and make them release their iron. And in the case of transferrin, when bound to a catecholamine hormone, when that iron is released, it can also be reduced into ferrous iron. Bacteria that are present in the blood use this relationship to steal some of that free iron that is released from these carriers from the host. So, when a catecholamine binds to transferrin in times of stress and it releases that iron to be used by other cells within the body, bacteria can send out their own mechanisms, such as siderophores, to grab that iron before it is used by the body and use it for itself. So, just as I said, siderophores can be used to bind to ferric iron when it is released in this manner, but also, as I said, catecholamines can reduce ferric iron when it releases it to ferrous iron, and siderophores don't bind very well to ferrous iron. So this 2+ form that is now floating freely within the blood serum can be brought into the cytosol similarly to gram-negative through permease channels within the cell wall. The 2+ form can diffuse slightly into the cell wall itself, but not all the way. So there are permease proteins deep within the cell wall that can bind to that ferric iron, or ferrous iron, sorry, and bring it into the cytosol through a channel to be used by the cell. So, those were just some mechanisms used by bacteria to take iron from the environment that it is in, pass it through its cell wall or cell membranes into the cytosol to be utilized. There is quite a bit of overlap between these two. Transferrin and lactoferrin are not used specifically by gram-positive bacteria. Gram-negative bacteria also utilize that relationship, but there's a bit of overlap, some differences in the specific mechanisms, but it is all to get iron into the cell to use to keep things. Thank you very much.